That's right.
Okay, so let's start. So thank you again for giving me the opportunity to present this work. I'm very excited to present here at SBDM. So I will talk about brain lesions in humans and effort and value-based decision making. So what's the motivation behind this work? So you may know that neurological disorders are the first cause of disability worldwide, and disability after a stroke or a brain injury is more often a behavioral disability than a physical disability. And for example, after a stroke or a brain injury, you can have many different behavioral disorders such as apathy, impulsivity, depression, anxiety, etc., etc. And the problem is that, except for depression, perhaps, we have very little treatment strategies for these disorders. And the reason for that is that we don't really understand the cognitive mechanism behind those disorders. So in this talk, I will try to focus mainly on apathy and impulsivity. So how could we try to characterize apathetic or impulsive behaviors in patients in the lab? So for instance, we could use these two kinds of tasks, willingness to work task or willingness to wait task. So for example, in the willingness to work kind of task, you have to choose if you have to decide if you're willing to make an effort in order to get a reward. And this kind of task has implicated in different brain regions, such as the insula, the VMPFC, the ventral striatum, or the dorsal anterior cingulate. And these regions seems to encode the value of the goal you're trying to reach, the cost of the effort, and sometimes the net value of the action, so the difference between the benefit and the cost. And on the right, in intertemporal choice task or in will task, you have to choose between a small reward now or a greater reward in the future. And these tasks have implicated the VMPFC that encodes the discounted value of the options, but also the DLPFC that seems to implement self-control, and also the hippocampus that seems to be required when you need to imagine a reward in the future and have this mental time travel. So these kind of cognitive models rely mostly on fMRI data, and we don't really know what the causal contribution of all these brain region is on these cognitive models. And we also don't really know whether these models are relevant for patients with brain lesions. So these are the two aims of the two studies I'm going to present. Is it possible to decompose behavioral disorders in patients with brain lesions in more elementary dysfunctions? For example, can you be apathetic for different reasons? Can you have a lower sensitivity to reward or a higher sensitivity to effort? Or perhaps that the weight of the net value is decreased in your decision? And secondly, can we use these brain lesions to test the causal relationship between lesion location and these elementary dysfunctions? So this is how we tried to do it. So we studied a large cohort of stroke patients, so 149 acute stroke patients. And so the advantage of studying stroke is that you can have very precise lesion boundaries, contrary to what you can have, for instance, in traumatic brain injury. And the second point is that in the acute phase of stroke, there is basically no chronic adaptation that could bias the brain behavior mapping. So we used the following design. So we had 4 main tasks. So the first task was a reward choice task. So the goal was to choose the best of 2 rewards. And the high value option and the low value option were defined based on the ratings of a control population in a pilot study. And so the second task was an intertemporal choice task, choosing between a small reward now or a greater reward but in the future. The third task was an effort choice task. So the goal was to choose the lesser of 2 efforts, and there could be cognitive efforts or physical effort, for instance. And the last task was a willingness to work task. So again, you have to decide if you're willing to make an effort to get a reward or not. and to try to identify brain regions that would be necessary to imagine an outcome in the present or in the future, we also added a second condition, an imagination condition, in which one of the two stimuli was only displayed as a text and not as a picture. So we used a single computational model to account for the choices in the 4 tasks. So what you have here is a generic formula of the value function. So this is the formula for the net value function. So this is a difference between the reward value Ri and the effort cost Ei. And when the reward was in the future, it was discounted by this hyperbolic function. And as you can see, the model has 3 parameters to set the weight of the reward, the effort, the delay, and the net value in the decision. And the choice probability was given by a softmax function, but as you can see, there is no stochasticity parameter, no beta parameter in the softmax. And this is because it's mathematically equivalent to the Kn parameter that sets the weight of the net value in the decision. So we fitted this model on the choices in all 4 tasks. And as you can see, the parameter recovery analysis was correct. So given the model, the prediction we made were that lesions in a region that encode rewards would decrease choice consistency for reward specifically, and would also decrease the willingness to work. Whereas lesion in a region that encodes effort would decrease choice consistency for effort, but would increase the willingness to work. And lesions in a region that encodes the net value between the benefit of the reward and the cost of the effort would decrease the choice consistency in all conditions and would decrease the willingness to work. And finally, lesions in the DLPFC and in the hippocampus would respectively decrease the willingness to wait and decrease the choice consistency, but specifically for imagined outcomes. So at the behavioral level, we found that on average, stroke patients had a lower performance compared to controls, and at the group level, it was significant for willingness to work and willingness to wait. So stroke patients were more apathetic in their choices and also more impulsive. And at the computational level, we found that stroke patients had a lower weight for reward and also lower weight for the net value. There was no significant difference for the weight of effort, and stroke patients had an increased weight for the delay, so an increased discounting rate for rewards. So this is what we have at the population level on average, but obviously there was a huge heterogeneity across participants and especially across patients. And so we use this heterogeneity to study the neural correlates of these different aspects. So we made a voxel-based lesion mapping study. So it looks like fMRI, but it's not fMRI, it's VLSM. So what you have on the top row here is the overlap of all the lesions of the group of patients, covering most parts of the brain, not all parts, but most parts. And the 2 main results were that we found a double dissociation with lower willingness to work being associated with lesions in the dorsal anterior cingulate, but these lesions were not associated with a lower willingness to wait. And on the other hand, lesions in the DLPFC were associated with a lower willingness to wait, but not with a lower willingness to work. So we also looked at the association with poorer performance in the reward choice and the effort choice. And again, we found an association with lesion close to the dorsal anterior cingulate in both cases. We then looked at the contrast between imagined outcomes and outcomes, and we did not find a region that would be associated with a general impairment in imagination, but instead we found category-specific association. So, for example, a lesion in the ventral striatum were associated with a poorer performance for imagined rewards compared to observed rewards. Lesions in the anterior insula were associated with poor performance for imagined efforts. And in the intertemporal choice task, where you have to imagine the reward in the future and make this mental time travel, well, a poorer performance for imagined reward was associated in particular with lesion in the hippocampus. So finally, we checked that the computational parameters of our model were associated with consistent regions in the brain, and indeed we found that a lower weight for reward and also a lower weight on the net value were associated with lesion close to the DACC. A higher weight for the delay was associated with lesion in the DLPFC, and a higher weight, so we also found that a higher weight, a higher sensitivity to effort was associated with lesion in the dorsal striatum. So finally, for this first study, we checked whether these objective measures in choice tasks, so willingness to work and willingness to wait, whether they had any clinical value, and in particular in predicting clinical outcomes. And indeed, we found that both willingness to work and willingness to wait, evaluated at the acute stage, predicted significantly disability just after the hospitalization. And we also found a dissociation with willingness to work only, but not willingness to work, being a significant predictor of disability at 3 months, and also a significant predictor of functional recovery during the first year. So on the right, what you have here is the evolution of stroke severity in 2 groups, and in red, as you can see, the group of patients with a low willingness to work had a poorer functional recovery compared to the other group. And all these effects were above and beyond the effect of traditional clinical predictors such as age, initial stroke severity, depression, etc., etc. So the take-home messages for this first study are that both willingness to work and willingness to wait have an interesting prognostic value, and the VLSM study confirmed the role, the critical role of the DACC and the DLPFC in setting the weight of the net value and the delay in the decision. But no, you may wonder, what about the VMPFC? Because we did not identify the VMPFC in this study, and obviously the VMPFC has a clear role in reward processing. So the reason that for that is that only very few patients had a lesion in that area in our cohort, and this is because there is a redundancy in the blood supply in this area, such that having an ischemic stroke in that area is quite unlikely. So we made a second study specifically to study the effect of lesions in the VMPFC. So what do we know about the effect of lesion in the VMPFC in effort and value-based decision-making? So there are reports, for example, on the left, showing that lesion in the VMPFC can decrease the sensitivity to reward, especially during incentive motivation tasks. But there are also reports showing that humans with lesion in the VMPFC can make simple choices between 2 rewards at a control level with no impairment. And on the right, during intertemporal choices, there are studies showing that lesions in the VMPFC increase the temporal discounting for rewards, and other studies showing no effects after lesions in the VMPFC. So the effect of lesions in the VMPFC is actually not so clear so far. And we thought one potential explanation might be that there are confounding factors that we need to take into account. And in particular, in many of these studies, most of the patients with lesions in the VMPFC were patients who had bleeding due to an aneurysm of the anterior communicating artery. So this is the anterior communicating artery. I will call this artery ACOM artery, and these patients ACOM patients for now. And this artery is just between the left and the right VMPFC. So it can create large lesions in the VMPFC and sometimes in the DACC. And usually when we compare these patients to a group of controls, we usually assume that any behavioral change is a result of these large lesions. But we also know that these patients very quickly develop a cortical atrophy and a subcortical atrophy, which is a consequence of the high intracranial pressure at the acute stage of the disease. And they also very often need surgery, so they have external ventricular drains with lesions in the cortex and also the white matter. And we don't really know what the contribution of these different lesions type is on their behavioral deficits. So to try to overcome these difficulties, we studied a group of 33 ACOM patients. As you can see, 11 of them had large lesions, including the VMPFC, and 22 had no large lesions, but obviously when you compare these patients to a group of controls, they have a widespread atrophy, at least in the ventral part of the frontal lobe and also in the dorsolateral part. And all these ACOM patients also have ventricular drains, lesions, and you can see in red all the entry points in the frontal lobe, and in green all the trajectories of these drains. So we tried to assess the effect of all these different lesions in effort and value-based decision making. So we used a very similar design compared to the first study with 4 main tasks: maximizing reward task, a minimizing effort task, a willingness to work task, and a willingness to wait task. And the main conclusion was that we found no significant difference between the 3 groups when making a choice between 2 rewards or when making a choice between 2 efforts. But we found a lower willingness to work in the group of ACOM patients with lesions in the VMPFC. And we have the a priori hypothesis that this effect would be driven by a decreased sensitivity to reward. And surprisingly, it's not what we found. We found no difference in value, in subjective value ratings. But we found that this effect was driven by an increased sensitivity to effort in the group of ACOM patients in the VMPFC. Finally, we found a lower willingness to wait in both groups of ACOM patients compared to controls, but with no difference between the group of ACOM patients with or without lesions. So the presence or absence of lesions in the VMPFC could not explain this lower willingness to wait. So we also tested whether it was associated with the ventricular drains, location, the side or the frontal, in the frontal lobe or in the parietal lobe, and we found no significant association. But finally, we found that across the whole group of ACOM patients, lower willingness to wait was associated with a greater atrophy in the GLPFC. So the take-home messages for this study are that we found no evidence that lesions in the VMPFC increase the temporal discounting of reward, but instead we found that these lesions decrease the willingness to work, and surprisingly it was driven by an increased sensitivity to effort, not by a decreased sensitivity to reward. The most likely explanation for the lower willingness to wait in these patients was the greater atrophy in the DLPFC. And these studies are an example of how brain lesions could be useful to understand causality in these cognitive models. But in return, it also shows how these cognitive models can be very helpful to try to decompose behavioral disorders in these patients with brain lesions into more elementary dysfunctions. So I'd like to thank the Motivation Brain Behavior team here in Paris, and especially Mathias Pessiglione, and all the students who participated to this project, especially Marina. I need to thank the stroke unit of the Pitié-Salpêtrière Hospital, and especially Yves Sanson, and also my funders, the Fondation pour la Recherche Médicale et la Fondation pour la Recherche sur les AVC. And I'd like to thank you very much again for your attention. Thank you very much.
Thank you, Raphaël, for this excellent talk. So we have one question from Miriam Kleinflüger. It's about the model you're using for delay discounting. So you use the hyperbolic model. Have you checked that this fits the data best?
Actually, no, it's a very good question. We could make a model comparison to check whether other discounting model would fit the data best. That's a good suggestion.
Another question by Nicolas Cléris. Were your initial predictions on each parameter, KR, KE, KN, and KD, confirmed? Yes. Yeah.
So that's a very good question. So not entirely. So for example, we did not find correlates of lower sensitivity to reward in the VMPFC. We believe this is due to the fact that we have very few patients with lesions in the VMPFC in our cohort, and as I said, this is due to the fact that having an ischemic stroke in this area is quite unlikely. For the other parameters, so we expected that lesions in the anterior insula and perhaps the DACC, but mostly the anterior insula, would decrease the sensitivity to effort. And this is not what we found. We only identified the anterior insula in the contrast when we contrasted the performance between imagined efforts and observed efforts. So we believe this still suggests that the anterior insula is implicated in evaluating the cost of the effort. But what happens is probably that the condition, the imagination condition, is probably more sensitive than the observed condition. So this might be why we just observe this, we just identify the anterior insula in this particular contrast. And I think for the other prediction, that was we basically found what we expected. Yeah.
Thank you. I might take one last short question that you may answer briefly, and then we'll turn to Pierre's talk. So the question is about the difference between cognitive and physical effort. Would you expect that if you compare cognitive and physical effort, they would reveal similar results in your first study?
So it's a good suggestion. We did not look at this difference. So we have indeed half of the stimuli are cognitive and half of the stimuli are physical efforts. We haven't checked that. I don't know what I would expect for this particular contrast. So I think in the model, the parameter capture the cost of the effort. I think it would be shared between cognitive and physical efforts. So we would probably expect the same region for the cost of these physical and cognitive efforts. Yes.
Okay, thank you. I'm sorry for the other questions. We might have a second chance at the end of the talks when we have the general discussion together. Otherwise, feel free to contact Raphaël and have a private discussion.
We'd be very happy to. Yeah.
Thank you again, Raphaël.
Thank you very much. Thanks.
Pierre, are you ready to share your screen and start your presentation?